All right. So do you think I should start? Okay. All right. All right. So let me share this. The okay. Share. And allow. Let's see. Let me go to slideshow. Ah, okay. Could you see and hear everything? Yes. Okay. All right. So since we are at the evening and after, I guess, after dinner, everybody, so we have plenty of time to discuss. I'm going to show you the recent work, and in a way, I hope that we everybody can discuss and understand. So I don't go to specific data. I just give you an overview, and I thought it is going to be interesting. Okay, so again, this is a PEG meeting for molecular subcommittee. I call it CORE. The title of my talk is Developing Antibodies to Polycystin 1 Proteins. Uh, okay, so I'm give you outlines what I'm going to do today. So I'm going to give you an overview of our subcommittee and define its purpose and the structure, how we operate. And then I'm going to discuss about why it matters, the work we do. And that is we develop and provides critical molecular resources. I'm going to tell you what they are. And I think these resources will drive ADPKD research, basic research. And then the end, and then the third part, I'm going to tell you about some of our key accomplishments. The tool we developed and the reagents we basically distributed to other people, to other investigator, and support how we supporting them during the funding period, which is the last 5 years. And by the way, if you have questions, just stop me anytime, and we can go over. Yeah. Okay. I'm gonna start with a very quick overview what we are working on overall. Now here is really a normal kidney. We have two of them, and down below, down below show you basically one of the million of units within the kidney who does the job. They call it nephron or renal tubule. This is a tubule. It's basically almost like a plumbing system in a house. Yeah. So this is a tubule where the blood is filtered in the beginning of the tubule. Yeah. And then they go through lots of segments, starting with the proximal tubule and then go to loop of Henle. This is actually a big loop. And then go back and then distal tubule, and then eventually they fuse or go together, merge into the collecting duct. And this is where the urine finally is refined and made. Yeah, and we have millions of them sitting within the kidney, and they do the job because you have a very sophisticated structure in a normal kidney. And this normal kidney is, of course, is disrupted, destroyed in case of polycystic kidney disease, PKD, by genetics. I'm going to go now, yeah, because the reason for that is genetic mutation. And genetically speaking, before we know anything about the gene and protein, people already know at the time when I start to work on PKD, we know there is a two type. One is called ADPKD, called autosomal dominant form PKD. Now, this disease is in contrast with the other form called ARPKD. This is actually affecting kids mostly. Yeah, I heard this is actually pediatric disease. Now, these two diseases are related, but they are not the same because the cause of the disease are different. In ADPKD, they're caused mostly by two genes. One is called PKD1. This is the name of the gene, and they're making a protein called polycystin 1, PC1. So I will come back to the name PC1. Yeah, and the other gene is less common. It's called PKD2. Protein is called polycystin 2, and PC2 is the short form. And ARPKD is caused by mutation primarily in one gene, is PKHD1. And the protein made of by this gene is called fibrocystin or FPC. I'll give you one picture to illustrate the differences clinically. So this is actually ADPKD. So in the ADPKD, you see already, as illustrated in the diagram right here, is that you see lots of bubble, as like a grape. Yeah. So this is actually focal. So they come from one cell. It grew in a focal fashion. Yeah. So as a result, you're going to see at the surface or within the kidney, you see bubbles. We call it cysts, of course. They're multiple. thousands of them each kidney. Yeah, and that drive the kidney to enlarge and lose the function, and the normal structure as you see in the normal kidney is destroyed. So the kidney fail to function. Yeah. So the bottom line is that they are focal, and so this is the basically main characteristic in ADPKD. But ARPKD is a little bit different. ARPKD, you see mostly affect the distal tubule. I mean the collecting duct. I mean the later part of the nephron, right? So instead of focal, the grade, they don't have a grade. Instead, the whole plumbing system blow up, became thicker and thicker. Yeah. So it's a different molecule, different gene, and different way of making PKD. But this is the three gene, PKD1, PKD2, and fibrocystin, or PKD1. Yeah. Well, I call it collectively PKD protein or PKD gene because this is the three major gene affecting most of the PKD patients. So the purpose or the very objective of the molecular subcommittee or core is really we want to advance basic PKD research. In our branch, and compared to other branches or other subcommittee, is to advance basic PKD research by a specific way, and that is by developing advanced molecular tools. And when we're talking about molecular tool, I'm really talking about antibodies and vectors. Yeah, I will come back to it, what they are. Yeah. For the three proteins, PC1, PC2, FBC, collectively, again, PKD proteins. This is one thing we do, our core do. Yeah. The second thing, which is very important, is to provide expert consultation. Because even if you get those good reagents, but you may not know how to use it optimally. Yeah. So optimally, yeah. So we are not only providing the stuff, but we also tell them how to use it, how to interpret that. So I call it expert consultation or technical service, basically. Yeah, we do two things. Okay, so this is basically overarching goal. Because we believe by providing those basic tool, you can do good research. And now the structure. Yeah, we call it molecular subcommittee. Now the chair is me, Feng Chen, UMB, University of Maryland, Baltimore. And co-chair is Robin Mazur, is University of Kansas, where Corey is. Yeah, okay, so when our U54 was funded, uh, actually my core originally at UMB called antibody and vector core. And it turns out that the whole U54, I'm actually am the only core doing this kind of job. Unlike other core, if you're gonna hear them, yeah, like a mouse core or cell core. I mean, many university, I mean, all three university had a similar core. They merged and became a subcommittee combined, but not so for me. I was the only person, yeah, does this kind of job. And the name with the red color are the people in my lab, either previous one in the old, I mean, before or currently was working on that inline core. But when the core U54 was established, was basically selected to work on U54, and the leadership, that is the Terry Watnick and Brad Yoder, thought that we have to do a subcommittee, not just me as self, lonely, you know. So they want me to have all other people from other two university to make a subcommittee, and so that I can get support from them. And I thought it was a great idea so that I can work with the idea with input of many other people. And then we're going to, and when this subcommittee was established, so we have additional member from our side. Yong Yu is the person come from St. John University, was part of my Iron Core. And then Patricia and the Terry of UNB joined my group in UNB. And these are the people come from Kansas. Robin is right there, you see, and then also UAB come with it. So other words, the three location together form the subcommittee, and now we call it molecular core or molecular subcommittee. Again, based on our original antibody and vector core at UNB. So this is how this core is actually established. Okay, so molecular core, we're interacting with research base through two things, antibody and vector. We make them and we give them. And then, as I said, we do technical consultation to them. And we have, of course, also very extensive collaboration within PKD RRC because we have other subcommittee. For example, we have cell model we actually working closely with them. We got material from them, which is help us to characterize and validate our antibody or vector. We also work with the clinical core and give us the patient material or give us the phenotype, I mean, how the kidney look like so that we can characterize our antibody on a clinical level. In vivo and preclinical, we have all interaction. I was actually a member of the two other core, cell model and preclinical core. Preclinical, I'm actually a member and actually partake their meetings. Okay, so this is so far the structure and purpose. Now, I said we do antibody and vector. So for the sake of time today, I'm just going to focus on antibody. And I hope that you will invite me back to talk about vector because this is a very big topic. Yeah, antibody is the one which I thought right now is most exciting one. So I'm going to focus on that. So before we start what we do, I'm going to give you an overview. What is an antibody anyway? Yeah, everybody talk about it. But in the reality is actually pretty simple. This is actually a immune system response. So antibody is something we as a human or any other individual, an animal, yeah, mouse, rabbit, no, any animal, they all have that. And why do they have that? Because they need to have this response in order to defend foreign invader and otherwise we're going to die. Yeah, but if foreign invader come, the body has this immune system. Yeah, in response to foreign invader. Yeah, so the way the body respond to a foreign invader is to produce antibody. This is a natural system in our body. Every single person of us has the capacity. They are produced, and why they produce? Well, they produce that allow the body to use this antibody to bind specifically to these invaders. And binding of those invaders will make them to be destroyed by other immune system. They're working together. So the antibody is really a what do you call it, is a sentinel. Yeah. And once the enemy come, they make the antibody protein and they bind to it and telling other people of the immune system to be mobilized to fight against invader. So this is how usually works. Now, in research, we take advantage of that system, the idea of the body's response. We actually make the antibody, but not like the body responds to invader. We do that deliberately. We can actually trigger a rabbit to produce antibody if we provide inject, for example, a specific antibody corresponding to the target protein, for example, PKD1 or PC2 or PC. And we call this particular antibody, a protein we introduce to the animal to make the antibody, trigger the antibody production. This is not invader. This is not a virus. Yeah, but this is to animal. They thought this is something foreign. Yeah, they make antibody against that. And the stuff, the protein we inject, we trigger the response. We call it antigen. Okay, so we use the antigen to produce deliberately the antibody in the animal. And we can purify those animal, isolate those animal, and we can use this artificial antibody to use in research. And because this antibody can function as like a sticky tags that will attack and label target protein instead of they target a virus. You know, our case, we just target a protein. Yeah, the body doesn't know. Yeah, they say, well, this is foreign. I'm going to make something against that. So this is basically looking like a sticky end. For example, this one is specific to PC1. This is PKD1 antibody, but I can inject PKD2 and proteins make antibody to PKD2 artificially. And we take advantage of the property of this antibody protein to do research. So that will bring us to ask the question, why do we need PKD antibody, i.e. antibody against either PKD1, PC1, PC2, or FPC? So again, we make those protein. And what do you do in research? Well, we can, number one, detect PKD1 proteins. We can ask the question, is this protein there or not there? Sometimes there, sometimes not there. So we need to ask those questions. And we use a technique called the Western blotting. We don't have to, I'm going to show you one example later on. But basically, this is one way to detect a protein, Western blotting. Yes or no, yeah. But then you can also to ask the question, but okay, but where are they? Not only yes or no, but you want to ask, are they at the in this cell at the surface or in the middle of the cell, or where are they? Yeah, so we can track PKD1 protein's location using a technology called immunofluorescence. Yeah, I'm going to show you one example as well. And thirdly, we can use this antibody to isolate PKD1 protein from tissue using technique called immunoprecipitation. Yeah. So and then you can use the antibody to study PKD1 function. Yeah. So other words, you can use those tag or antibody to ask the question to study how this PC1 or PC2 or FPC, how they look like, how big they are, where they are, and you can isolate them to asking what other partner do they talking to. Yeah. So all these basic questions we can answer using this antibody. And this is a very, very useful tool. It's basic tool. If you want to study proteins function, what they do in the kidney and why do they form, if dysfunctional, making a cyst, you need those antibody to answer in the laboratory. Uh, is there anything you have a question about that? Let me see. Because I don't. Okay. Yeah. Okay. You can. Yeah. You go ahead.
Yeah. Okay. Hi. Yeah, so is this imagine I'm a researcher. Do I get just some cells, some antibodies like actually shipped to me, or is it or is it put into an animal, or how is it delivered?
Oh, yes, that's a good question. Yeah. I will actually give you example, but basically, as I said, we produce antibody deliberately, and the antibody is made actually in the animal. People make it too, but this is hard to ask people to make it. So animal, you can actually inject your protein, say PKD1 protein antigen into animal. Animal see that protein coming into my body, thinking this is something bad, and they could
Mhm.
defend themselves against this PKD1 antigen. And they do so by producing an antibody against that invader, so-called invader, but in reality is our PKD1 antigen. And we can get the serum, get the blood from the animal, and we can isolate the antibody. And my job in our core is really to show that this antibody, this particular rabbit or mouse or rat, there's a different way to do it. Yeah, many animal can do that. Yeah, but once you get it from them, you want to make sure that the antibody this animal make good antibody because there's also bad antibody. Yeah. Now I will tell you what is good antibody, and we actually have a way to tell is good antibody. Now, when we have that, it's delivered by typically a company. We're going to do that for you, but some university has their own rabbit in the facility. You can inject by yourself, and you can get the blood, and you can test and wait different time, and because they need time to develop those antibody. But at the peak time, then you can get those blood, and you're basically using technique to purify the specific antibody, and we're going to test that in the lab. And now once we have it, and we're going to tell the people in our website to say, hey, we have a very good antibody you can use to study PKD1 in a kidney, normal kidney compared to a PKD kidney to see how a dysfunctional or mutant protein look like. Are they looking differently than a normal protein? Maybe they go to different places, or they are processed. They are modified. That, in other words, for a protein to get really working, functional, they undergo multiple changes. Like a butterfly. I mean, you start with a one form, and they have to go through different phases until they are really, really good. Yeah. Maybe, I mean, in PKD1 patient, yeah, sometimes we see that this so-called modification, that's the name of changing, yeah, towards maturity, they are stuck. So therefore, the protein may be made, but they are not functional as a mature protein would do. And this antibody allow us to actually to assess which step this mutant protein is stuck with. And each family, because mutation PKD1 is so different, and the effect on the protein is also very different. Some make the protein completely disappear, and we can use the antibody to test whether they are there or not. Some you don't see any protein anymore. But sometimes a protein is made, but they're looking funny. They look differently. Yeah, and these are the question we can actually answer using our antibody. Yeah, and once we know that, for example, if you want to develop a therapy, you need to know the reason of that particular patient or mutant. right? So if you don't have any protein, then you cannot make a drug to change the affinity, activity of the protein because the protein is gone. As opposed to a family, when you have a mutation, we know using this kit, you can using our antibody, you can say, this antibody missing an important step in maturation. This antibody can tell. If that's the case, we know that we could potentially use a drug which help the mutant protein to mature with help of a drug. That will make them functional. So that's really important for if you think about in the realm of therapy. So this antibody could allow the researcher to answer basic in the lab the question before we go to the clinic. So basically, our lab molecular tool is really, first of all, in the lab of basic research in terms of the property of this key protein, PKD1 protein, and what kind of defect it could actually undergo, and how maybe in the lab we can actually correct that. And the antibody is going to be used to detect efficacy of any treatment because you need to know whether the maturation is restored, and our antibody could potentially do that. And once this tool is established, then other people or even my lab could thinking about a drug. Then you add it and you see, I can see this antibody can tell us whether my drug has an effect on the PKD1 protein level. So in terms of
Yep.
delivery is really onto the lab. It's not going to the patient. This is not a drug per se, but it is a tool to help us to understand how the protein behave or misbehave, and hopefully have a way to use it to assess efficacy of any drug we have.
Yes, thank you.
Yeah, great. So any questions if I'm not sure, because I try to do it in a way that I hope that you could follow me, not using those fancy scientific terms.
So, Fang, if I
Yeah,
can,
yeah.
if you could explain to them how the antibody is actually prepared and then shipped and how it's used, because
Oh,
when you actually ship it to them, it comes in a vial and it's like inside of a
oh,
solution.
oh,
So I think that might help too for them to understand
oh,
how
yeah,
it actually how the scientists actually use it.
yeah. Okay, so we make the protein, and we the protein. I mean, the antibody is in a liquid form. This is pure protein. This is basically a protein. Protein. It's like yeah, any protein like egg protein and albumin. This is like those protein. Yeah, but this is just immune protein, and we resolve the protein in a salty water, and we make them in small aliquots, make them in small portion. And if somebody in another lab, say in San Francisco, University of, let's say, Pennsylvania, right, where Lisa's working. So if she wanted to have antibody, she can put a request in our website to say, I need to have antibody this and this because Dr. Chen has developed it and they have such and property. I'll give you one example. And I will actually pack them and just ship them by FedEx in a tube form on the dry ice. And they will be delivered to their lab and they put into the freezer. And when they have it, they can use the reagents in different way, depending on whether you're going to do Western blotting like this, or immunofluorescence or immunoprecipitation. Yeah, this is a different way of application. Yeah, and then going to take an aliquot of an antibody, and I'm going to tell them how do you use it, what dilution you're going to do, and how would you analyze the result. Yeah, this is my technical consultation part. Yeah, I will maybe get to real picture about it. Okay, now we do have some existing PKD antibody. Yeah, it's not just we don't have it. Yeah, this is just coming from our core. You see, you have PKD1, PKD2, and fibrocystin. And each small black bar, you see, this represents the pre-existing antibody. We made it 10 years ago. We have been in the previous funding cycle, and we actually keep supply to the investigator. Yeah. The problem with that is that at that time, we used a technique, you were using fragments, small pieces of antigen, as I said, antigen, inject either into rat or in a rabbit. Yeah. So the quality was not good because this is small fragment. They usually made in E. coli, believe it or not, E. coli. So the protein we made is linear. Yeah, we call it denatured. So they are not in a native form. Yeah, because E. coli do not produce a native form. They usually get denatured, like an egg, you boiled them, so they became hard, denatured. So we use an antibody, an antigen producing E. coli, they are denatured, they looking not the same as in our body. And so the antibody made against those type of antigen, they are limited in the scope. Sometimes they cannot recognize the native protein in our body. So the potency is not very high. They're good for something, but not good for everything. And this is a limitation for the field. And that is particularly for PKD1 is very challenging. Think about this way, like a Waldo, it is in a crowded area. Everybody represents one protein in the cell, very crowded. But to find PKD1 is very difficult and that's particularly for PKD1 because they are very large. Large proteins very difficult to detect, and they in low abundance. That means a very few molecules present in a body. So it is very hard to catch because they are hidden. There are only a few, and they are membrane protein. That means they're sitting at the surface of a cell. Now this is a location our techniques usually have hard time to detect. If you are in the inside of much tends to be much easier. And then these protein undergo additional cutting cleavage. So you make a big protein, fine, but eventually the cell chop them in small pieces, and that make it detection even more difficult. So in other words, this is very challenging. Although we have lots of antibody, but we're made in a way that is not optimal. And on top of that, and the protein intrinsically is very, very difficult to study anyway. So with that, people really have very hard time to detect PKD1 in any tissue. And I say we actually published a paper to say many of the antibody published in the last 3 decades, I would say many, if not most of them, are non-specific. They're wrong. So we really have a good antibody to see what happened in the body. So this is a challenge. So therefore, but the PKD1 is very important. It is encoded by PKD1, is the most common gene mutated in PKD, and so therefore is the principal protein associated with PKD1. And PKD1 is the key driver of cyst formation in both the kidney and liver in PKD. And liver disease as well. So PKD1 has more role, not only in our PKD, but also in the liver disease. This PKD1 has, and PKD1 is important. So therefore, defining PKD1, PC1 function is really essential to understand disease mechanisms. So we are focused on PKD1. So now our core has this particular goal, 2 goals, but this is the goal, the major goal is this, really to develop new, high quality, reliable PC1 antibodies as a tool to detect native PKD1 in our tissue and cell. And once we have it, we can make it accessible to the entire research community because other people are not going to do it. And I was chosen by the U54 to do that thing because I have long-standing interest and we did lots of success in the other type of antibody. So we are committed really to say, okay, I'm going to just put our own effort in my core to really develop a different kind of antibody and hopefully overcoming the previous obstacle and making really great antibody. So this is our goal. But what do we do differently? We cannot repeat the old-fashioned methods and hoping that the result is going to be different. That's the wrong approach. So therefore, we propose using completely different way to do it and hoping that different results are going to come. So we do 2 different new approaches. Number 1, we're going to making so-called rabbit monoclonal antibodies. Number 1, this antibody is made in the rabbit. Rabbit is by far the best animal to make any antibody. And therefore we chosen the right species, rabbit. But people use the rabbit all the time before, but it is the monoclonal version of rabbit antibody, which is new. Monoclonal means it comes from one single cell, just one single specific type. And instead of polyclonal, which people old-fashioned traditionally always make, that is to say that you shoot into the animal, animal is making lots of antibody, and each cell you can make a different kind of antibody. So and when you isolate those polyclonal antibody, they are not very clean. They're not very specific. But we make monoclonal, they're making very specific. So rabbit monoclonal, they have these following characteristic. Number 1, they have high affinity, means they bind to your target very, very tightly. Much tighter than a mouse antibody would do. And then they have high specificity. That means they have the affinity to recognize only the PKD1 antibody. They don't find anything else. Now, that's very important because if you have an antibody recognize PKD1, but as well as other protein, then this antibody is non-specific. You can have lots of non-specific signal you're going to create, make an antibody dirty and not useful. Yeah, but this rabbit monoclonal has the affinity, potentially have the potential to overcome this problem. So high affinities, high specificity, and this is very, very important for a good antibody. And they of course have a consistent performance and renewable source because it's a monoclonal. Yeah. For the technique, which I'm not going into it, but it is a clone. It is consistent. Each time you can have the same antibody again, all over again. If you make another time, you can make identical. Unlike a more polyclonal, each rabbit give you a different population of antibody. The second approach we do differently is we use a different kind of antigen. Instead of E. coli made, which I have said, we made a mammalian cell express antigen. So we ask a cell to make mammalian cell, to make this antigen for us. The reason for that is that those protein, which is made from mammalian cell, because it's much closer to us than an E. coli would do. So the protein antigen they made has natural protein folding. The protein is folded very nicely because, as I said, denatured protein is stretched like a linear thing. But in the reality in our body, the protein is folded, in a particular form. And when these mammalian cells make this antigen, they have this capability. Once the protein is made within the cell, the protein will actually fold very closely to the native protein in our body. And that is important because once you use this antibody, antigen, give that to a rabbit is making antibody against this folded protein, which look much more like our own native protein. So the antibody has just simply better quality because they're going to recognize a native protein as opposed to the previous approach, recognize a denatured protein, which doesn't like our native conformation. So in other words, this will make a better antibody, recognize endogenous protein in our body better. Okay, I'm going to make a stop for any other question you may have. All right. Okay, so we did differently. Now, to make a story short, I'm not going to go to specific steps, but basically this is what I achieved using these two strategies combined. Okay, now this is polycystin 1. I mean, this is a diagram looking what each domain, each region of the protein looking like that, and this is the PC1. And basically using this technology, we developed and validated 14 distinct PKD1 monoclonal antibody. RMMA, this is a rabbit monoclonal antibody. This is a short form, So we actually made it 14 of them. And they actually directed to diverse region, different region within PKD1. For example, we have A, B, C, D, F. Yeah. Now this is actually quite different. I mean, when we do antibody, you usually do only one. Just one antibody, that's it. And this is a big achievement if you have one. But in our project, the way we did it using this new technology, we did it at 14. And 14 is typically unbelievable. It's a huge, usually one, but we have 14, And they're directed to different regions. And each number represents one antibody. And they can be effective across diverse detection and application. As I said, we can use the antibody to even do like Western blot, immunofluorescence, immunoprecipitation. There's a different way to detect antibody, either location or the size. You can use it for a different reason, a different tool. Some tool is like a toolbox using different purposes. And this antibody can do for all of them. And they are across a different region. I think it has implication for detecting mutation in each individual patient. So I think if short form, we using this core, we actually, before we did one or two, but this time we did it for 14 of them. It's really huge, I would say, success. Now, this is just give you 3 example. We actually validated, we put it into a product looking like that. I mean, if you like Amazon, right? I mean, if you click a specification sheet, this is exactly like Amazon, So we posted this on the website. People can look them and to see how they're made and what's the region they are. how they're going to use, and this is a few example. I'm not going to go through this. If you want to look at it, you can actually, this is actually available in the website. But bottom line is that the three product is the best among the 14. Other 14 is also good, yeah. But this is the three we posted now because we don't have to post all 14. This is going to be confusing. They're not going to know. If you provide them too much, it's not good. So, but the three is the best. If they want to more, I talk to them. So people can say, I want to have 45, or other people say, I want to have number 7 because they go to a different region, and they can be used for different things, yeah. And people are going to talk to me, and I'm providing service to them to say, okay, what do you want? They say, I'm going to know whether the protein is made in this particular species, I mean, in this kidney. I say, okay, that if you want to use that, you can do 50, 45 is the best. Or other persons say, well, I'm going to want to know where the protein is within the kidney. I would say 7 is good, use that, but you can use 45 as well. So this is a product people can ask, and we actually are providing these reagents to various people. And this is basically a hit in the top 10. I mean, these are the three. Now I give you a little bit the example how they look like in the lab. I mean, I'm telling you only on the paper. Yeah, I'll give you one example. I want to know where the protein are. Yeah, normal kidney versus ADP kidney. I just give you just one shot. Yeah. Okay. Here you see, oh, what are they? Yeah. Look at the number 7. This is number 7, our antibody. They put into normal kidney of a human. Yeah, and provided by Cell Core, another core from University of Maryland. Yeah, and they provided me this thing, and we put the antibody in to see where is PC1 antibody present in the kidney. And we found, for example, those kind of, the ring, this is actually a kidney tubule. Yeah. But the question of what does it mean? So for answer that question, we actually can put another protein called LTL, green. And this is, we know LTL is a marker labeled the proximal tubule. This is actually this part. Yeah. So as you see here, some of the red thing co-stain with the green thing. You see some of them are stained together, other one not. So we can say that in a normal kidney, our PKD1 protein is present in this area, not the other area. Yeah. And subset of those. Now this is new. Previous antibody never saw that. We use different technology to prove this is real. So we think we have a breakthrough in a PKD1 antibody, doing things in the human kidney other people have not done before, because the antibody was no good. This is good. Yeah. And in ADPKD kidney, I didn't show you the signal was gone because they have mutant protein is not detected. The second example I'm going to show you, and this is exciting in the field, is the so-called Western blot. I'm showing you. How do you know how does a protein look like that? Yeah. Now this is a gel and looking like boring, but to me it's very exciting because it tells you, for example, this is NHK, is normal human kidney. Yeah. And if you put the antibody in this Western blot, it detects this band. We call it band. Yeah. And we know this molecular size. I mean, we can know how big that is. And we can see 2 bands. Yeah. And you cannot see very clearly. Yeah. But you can have 2 bands. And that means they're matured. And we know in a normal kidney, this is what you should be looking at it. Now, these 3 samples are come from cystic kidney. I just want to point out one kidney here. This one kidney, yeah, this is one of the 3. You see, the protein is not there in this place where they should be in normal, yeah. Instead, you see a protein right here. You see, this is the mutant protein. And what does it say is that the protein is run in different places in the gel. That means they have a different molecular weight. In other words, this is much smaller. This is a bigger protein. It should be like that. But since the protein has mutation, and the protein is much smaller. Yeah, so we know this patient has a truncation, we call it. Yeah, it's devastating mutation because the protein is much shorter. So that probably is non-functional. Second example I'm going to show you is we tested urinary exosome. Now this is interesting because there is a protein, there's a vesicle is produced in the tubule. Vesicle actually is emerge from the cell and they dump into the urine, and you can collect them. Urine, and you actually can prepare, isolate those exosomes. This is a vesicle. Yeah, we found that this vesicle could be contain lots of PKD1 protein using our antibody. Making possibility. I mean, we're thinking about to even make a kit. You just harvest the medium. I'm sorry, harvest the urine from a patient. around the gel like that, compared to the normal person. And I suspect that some of this patient, we're going to produce a different protein. Then I can tell, okay, this patient has a mutant form. Probably you could, in the future, you're going to use a targeted therapy specifically to this truncation protein. I'm just example. Now for the future, I think it's very important for us because possible application. I probably not going to go through it. Yeah, you can actually look at the slides. You can read it. Yeah. So basically, protein detection. Yeah, and then you can looking at the localization and the functional study. Yeah, particularly important is to me is exciting is maybe highlight this excess effects of specific mutations. Yeah, once you have genetic analysis, doctor may tell you, oh, you have a point mutation, but you not know whether this point mutation is disease causing or not. For example, I could using urine around the gel, I found the protein behave abnormally. I can basically with high confidence using this antibody to tell you, aha, the protein made uh, uh, but continue that particular mutation, although you cannot tell by genetics, by genomic sequencing, but using our kit, I may able to tell you either the protein is not made or they're made, but they're looking funny. I can tell you whether this is disease causing or not. And in the future, when a therapy is made, I can even tell you that you have this type of mutation and this particular drug may be more effective than another drug. So that's the value of, I think, this antibody. Because we have 14, we can make it potentially a kit utilize in the clinic. This is my dream. Yeah, my contribution. Yeah, the course contribution. Okay, so in the end, I will say why it matters, the work. Again, only antibody. The vector, I didn't even touch it. Next time we may talk about it. Well, why it matters? Because I believe that our work, antibody, new antibody, addresses a major bottleneck of highly specific PCD antibody, PC1 antibody in ADPKD research. So finally, we have, in other words, an antibody which we are confident that the antibody see the protein very reliably, as opposed to antibody which is dirty. You're never sure is this PKD1 or not. You see only bands, yeah, but bands is many bands, yeah. But I can tell this band is truly PKD1, nothing else. So this is a major, I think, is achievement. It matters a lot because then you have a tool to study your PKD1 protein in ADPKD research. And it helps researchers to understand PKD1 function and its role in cyst formation in ADPKD. This is what this PKD antibody for. And I would argue for the future is I think also very important because it could facilitate development of new treatment. Or we can say whether a treatment is effective, we can use our antibody to tell. And now finally, maybe the last second slides, I'm going to tell you, but okay, so I tell you everything development because this is the most exciting one, new things. But this is not only thing what we do. Yeah, we have accomplishment through during the last funding period using our resources, do the following thing. Number one, we have delivered over 175 reagents shipments worldwide. People asking us, I mean, we're too busy. I wish we have more hands. I mean, we can ship them anytime, yeah, we want. But now we have to wait a week, because otherwise the other people cannot do other work, yeah. So we actually have lots of requests, 175 shipment worldwide during our 4 years. Now we have even more when we count it now. This is November, when we wrote the grant, actually 175, yeah. Number two, we provide extensive video and phone consultation. This is mostly my job, yeah. And then number three, we contributed to 17 publication papers, advancing scientific knowledge using our reagents. And then lastly, we produce, now this is also, I'm very proud with our core, we produced the first educational video on PC1 processing and analysis. And this is posted already on our website. So people can watch it like a movie, yeah, to see how PPD1 look like and how we analyze them, how they process to maturity. I think summary. Yeah, summary, we developed and validated high-quality rabbit monoclonal antibody against PC1. And these are essential for studying the process, the protein across various applications. And we distributed reagents and provided support to investigators. Yeah. And next step, what we're going to do? Well, since we have this antibody, we'll make the antibody broadly available. Now the company also ask for us now. Yeah, many now. And we're gonna keep making those antibody because this monoclonal, we can make them again the same quality. So we're gonna make antibody broadly available. And number two, we're going to do is support collaborative studies since we know how to use it, and people now are asking more and more, and we're going to do collaborations. Number three, we're going to develop antibodies for other PKD-related and protein. So far, I said only PKD1, PC1, but we have PC2, FPC1. Still lots of room to develop antibody against them using our new approach. And I hope that this particular funding, the new funding period, will allow us to do that. And I'm going to stop here. I'm sorry, I'll take a little bit longer. But any question and advice, you're welcome to do. Let me get this. Yeah. Okay. So any questions or anything you want to ask me, I can discuss. I will be glad to do for you.
Hi,
Okay.
can I ask a question?
Yes.
I was just curious, like, how do you or get the word out that you've got this great antibody, that you developed this great antibody? How do you advertise it to researchers, or how do you let people know this is new, this is something different?
Great questions. Three things. Number one, we post this new edition in our website. We put it into the first page. And this is number one. Yeah. Number two, I go to different meetings, like our annual symposium, and they give us always each core or subcommittee 10 minutes time. I made it a video talk showing them the products and showing them some example. And I think people know, somehow they know we made a good antibody. And lastly, and this is something we try to do, white paper something, and we are actually writing this result in a paper form, writing a scientific paper. And we will almost done. Manuscript is written, but we have some improvement to do. We're going to submit it to a scientific journal, like Kidney International. This is our target. So that is published, and people may read it and to say, I need antibody, and say, oh, this guy have it. And they may come to our website to read it. And I think there's other way. Terry Watnick.
So yeah, Fang, I also send out like blast emails, and then when we have meetings, I also post and advertise the new antibodies, whatever it might be. If it's an antibody, if it's a mouse, if it's, some sort of vector. So it's advertised multiple ways by sending out emails and when we have meetings.
Yeah.
Thank you. It's a great talk and very exciting. Thank you.
Thank you. Susan.
This is the Slima. I have some basic questions.
Sure.
The antibodies you mentioned, and these are for PKD1.
Correct.
Do human being, because so far your research are with the animals, not the human beings. Am I right?
The research is done mostly on the animal, correct. But the antibody I just talked about, we made it, is against human.
Okay.
PC1.
So human beings, like those who do not have the PKD, like here it is PKD1, they do not have antibody, or they have the antibody of the PKD1 or whatever. So that's why they do not get the PKD. And what is the difference between the human beings, those who have PKD and those who do not have PKD?
Good question. Yeah. Okay. So this is how I'm going to see that from a research perspective.
Yes.
Okay. So PC1 or PKD1 protein is a normal protein.
Okay.
Everybody has.
Okay.
So a normal person without mutation, so without PKD, yeah, we do have PKD1 because PKD1 is needed by our body to prevent PKD1. Yeah.
Oh.
Now for PKD, yeah, so everybody has normal PKD1 protein in our body, in our kidney. And you need that because you need to use this protein to develop a good kidney and function probably. You need PKD1. This is the normal function of PKD1 in normal people.
Okay, so those who have the PKD disease, like the cyst developed, so they do not have the PKD antibody. Am I right?
Oh, no, antibody is different issue. I come to next step.
Okay.
I'm just first talking about a native condition. I mean, as it is, we have PKD1 protein, and if a PKD patient come and they inherited a mutation in the gene, so that means that this patient is making a defective PKD1. It had defect in it due to mutation.
Okay.
Because the mutation is not good for the protein because you're causing them to function abnormally. And as a result of that, you don't have the normal protein, and that person or that patient will miss that function. Now, as a result, PKD cyst is formed. PKD formed is because you don't have a good protein in it due to mutation. Which is what happened in the body. But how do we know? How do I know, right?
Exactly.
You know that because I have antibody to detect.
Okay.
Yeah. So the antibody is a tool for our research to find what's the normal PC1, PKD1 protein look like in our normal body. You need to know what they look normally.
Okay.
Then you can answer the question. If a patient come to me, I can say, you have a mutation and you have a dysfunctional protein. And the PKD1 is antibody is a tool which allow me to see the difference, to see the defect. This is a tool.
Okay. Okay. So for when you said that you have discovered 14 types of antibody,
Yes.
right? And how many more can be like an antibody? Is it and do you think if you can identify the antibodies that will make the kidney normal,
Okay.
can you like uh, put it in the human being who have PKD disease, okay, that antibody, and then their kidney can come to a like normal function kind
Yes.
of.
This is very interesting. You should work in our lab. Well, what you're really asking is this. I'm saying this is a tool. A tool is, cannot change the course of the disease. It just tell me what's wrong, right?
Exactly.
Yeah. But are you thinking about is the antibody has even therapeutic effect?
Yes.
Oh, yeah. I think it has a potential. Now, this is something our field, nobody talk about it, not even think about like you think. But I think the reason I think it could be possible, think about it. I think if I have a very, very, very good antibody, I know my antibody can bind PKD1 protein.
Okay.
How about that? If I actually couple this antibody with a drug, I insert that into the body, in the blood, in the kidney.
Yeah.
And the antibody is like a missile.
Yes.
To move around, find the target. The target is PKD1 protein.
Yeah.
Now, in the normal people, we don't need to do that because it's not going to be good because But in a PKD kidney, if the protein is made, but it's just a bad protein, and then I can use this antibody to let it go to find this mutant protein.
Yeah.
And if our mission is to destroy that mutant cell,
yes,
making cyst, I can actually couple a toxin into my antibody and destroy that particular cell. Some cancer drug is like that.
Yeah.
Right. But for that, we need more research to say, is this a good idea to destroy cystic cell? Some people say yes, because they thought cystic is like a benign cancer. Yeah. So you destroy this cyst. Too many cells, you can destroy them by that, But this requires basic research to see whether this is actually a good idea or not. But basically, is it therapy? I think is possible. You can even, another way to think about it is, I have the antibody bind to the mutant protein. Maybe the binding itself can make the mutant protein more active.
Yes. Yes.
Yeah, it is a possibility. Yeah. Now we have 14. We can make more. Maybe one of them just doing that job. We can test one by one in our lab. And one of the person in the subcommittee in John's University, I just asked him to do that just for that, to see what the antibody has a function altering function.
Exactly.
Yeah, exactly.
So then going back to your question that you need lots more research, how much more research you need, because based on your experience and how much funding you need now, all the fundings are cut or eliminated kind of. So how much more funding you need, and with the time, how much time you need, so you can see whether any of the antibodies can kill. I'm using the word kill because that may not be the right word for those who have the PKD disease, so you can modify it.
Yeah. So how much time or how much effort do you need to eliminate those mutant cells causing disease? In the animal, I mean, in this case, we can work first in the animal model to see it. Yeah. Now, this is, of course, I mean, I will say this, I mean, if I have a wish, unlimited funding, I have lots of projects in my head. I just cannot do it at the same time.
I'm sure you have.
Yeah. No, but the thing is how much? This is really, nobody asked me the question, how much. I would say, no, I mean, I say, but in the United States here, in the University of Maryland, so for example, in our case, we have like 2 people.
Oh.
But how about if I have 20 people? Now, other country, they have 20 people. There are 50 people in my group. I can work out all the conditions. Each person work on the different things. Exactly. I have to pick one and hoping that particular one is working, but I may be wrong. So I have to work with the best guesses. And so as you see, I mean, 20 times, that would be really good. Yeah, but I mean, this is really a thing. Yeah. For example, you need a good mouse model to test, right? Right now we have maybe 5, But I have some idea even. We can have a better model to do it. But this is basic a grant. I mean, to make a grant nowadays is cost really.
Yes, I understand that.
Yeah, then I'm retiring almost. Yeah. No, I'm just
don't
kidding. I really want to do it. Yeah. No, what I'm saying is this is limited. I mean, this is tough. I'm fighting every day to get the money, and hopefully this time this funding grant. I mean, this cycle, the new one. Apparently we have a good score, and we could continue to do the development, and also hope that other group, not only mine, but many other group, they have the tool. Maybe that's empower them to do the thing they want to do.
No, at least you provided the tool. So if others can, you identified 14 antibodies, and if you can, think of, okay, that many antibodies, probably can get 1 or 2 of them.
Yeah.
To, if you can call these antibodies as a missile to get into those diseased kidneys. So, and you said it is not just the kidney, it is also the liver. So that would be I understand that it is a lots of work, and it is not just going one or the other.
I have sent, for example, my reagents, I mean these 3 antibodies, to a group in Seattle who developed this organoid system. He can develop this basically a organ in dish.
Oh.
Yeah, he could do it. Yeah, he could. I couldn't, but he do it. But that's the reason we do collaboration, right? I could.
Yes, definitely.
Yeah. So I have already sent this few antibody to him, as I asked him to add the antibody and show me whether the antibody could alter the course of, let's say, cyst formation in our body. It can, but in the organoid system. Now I don't know what the results going to be, but this is something I'm reaching out to different people, telling them, here, this antibody, if you're working with the human system, this is good. This you may want to give a try. And we try to reach out if the funding allows. I think it will. To the UAB, because there's another group, they doing wonderful work with the human organoid. They can make cysts, they can make tubule, and I asked them, no, this is not. I mean, I have the idea. I'm going to suggest him to say, okay, use this antibody, that antibody in your system and show that you can cure the cyst at least in your system, right? I mean, that's a good first step. Yeah.
Thank you.
So exciting.
Okay.
Yeah,
Okay.
thank you so much. It's
Thank
really
you.
Sure.
That was great, Fang. I thank you for coming. And if anyone has any additional questions, please email them to myself or Fang, and I'll make sure that he gets it.
Yeah.
Okay.
Thank you very much.
Thank you
Sure.
so much.
Thank you.
Very
Thank you very
interesting.
much.
Exciting.
Bye-bye.
Bye.
Bye.
That sounds very